Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, I am now delighted to introduce our special guests of honor, whose presence today underscores the importance of this endeavor. Today, we are privileged to have with us an exceptional panel of leaders, experts, and visionaries who have made remarkable contributions in their respective fields. Allow me to introduce our esteemed special guests of honor. First, we are honored to welcome Professor Abraham Mitike, the Principal of the College of Medicine and Health Sciences, University of Rwanda. Professor Mitike's leadership in medical education and research has been a beacon of progress in the healthcare sector. Next, we have Dr. Eugene Mutimura, the Executive Secretary of the National Council for Science and Technology. Dr. Mutimura has been at the forefront of advancing science and technology policies that shape Rwanda's development. Joining us as well is Professor Claude Muvunyi, the Director General of Rwanda Biomedical Center. His dedication to strengthening public health systems has profoundly impacted the nation. We are also privileged to have Dr. Sekamo, the Director General of National Industrial Research and Development. Dr. Sekamo's work in fostering industrial innovation and development is truly commendable. Another distinguished guest is Professor Emile Bienvenu, the Director General of the Rwanda Food and Drug Agency. Professor Bienvenu's contributions to ensuring the safety and quality of food and drugs in Rwanda are invaluable. It is an honor to welcome Professor Leon Mutesa, a Professor of Human Genetics at the College of Medicine and Health Sciences, University of Rwanda. Professor Leon Mutesa's pioneering research in genetics continues to break new ground in understanding human health. We also have Dr. Dieudonné Mutangana, the head of the Department of Biological Science at CST University, Rwanda. Dr. Mutangana's expertise in biological sciences has been instrumental in academic and research excellence. Finally, we are delighted to introduce Professor Janine Kondo, the CEO of the Center for Impact, Innovation, and Capacity Building for Health Information Systems and Nutrition, CIICHI. Professor Kondo's leadership in health innovation and nutrition has made a lasting impact locally and globally. Please join me in giving a warm round of applause to our special guests of honor, whose presence graces this important seminar. Next, we will move on to Dr. Silvia Muñoz Descalzo, Quantitative Developmental Biologist, Profesora Contratada, Doctora I3, IUIBS, Universidad de las Palmas de Gran Canaria, Spain. Dr. Silvia Muñoz Descalzo is a renowned quantitative developmental biologist, Profesora Contratada Doctora at the University Institute for Biomedical and Health Research, Instituto Universitario de Investigación Biomédica y Sanitaria, IUIBS, at the Universidad de las Palmas de Gran Canaria, ULPGC, Spain. Dr. Muñoz Descalzo has an impressive portfolio, with 31 publications in leading journals, reflecting her profound contributions to developmental biology, cell biology, and genetics. Her work at the intersection of biology, mathematics, and computational modeling is centered on understanding how cells process and integrate signals to make critical decisions during development. Dr. Muñoz Descalzo's academic journey began with her PhD research on Drosophila, where she mastered genetic tools to study the impact of signal transduction on developmental processes. Her early focus on Drosophila models laid the foundation for her postdoctoral research at Cambridge, where she explored the intricate interactions between Notch and WNT, signaling pathways in cell fate determination on the imaginal disc. Driven to gain insights at the single cell level, she later transitioned her studies to mouse embryonic stem cells, MESCS, and early mouse embryos, where she revealed the essential role of beta-catenin in maintaining pluripotency. Collaborations with mathematicians and physicists produced one of the first theoretical models for beta-catenin's function in pluripotency, highlighting her commitment to interdisciplinary research. Throughout her career, Dr. Muñoz Descalzo has advanced key methodologies in developmental biology. She has been instrumental in developing and testing 3D segmentation tools for confocal imaging of early embryos, a tool that has transformed how cellular dynamics in embryogenesis are studied. Recently, her research has expanded to assess the impact of lifestyle factors, including age and maternal diet, on fertility and pre-implantation development, an area that translates fundamental science into actionable insights for human health. Dr. Muñoz Descalzo's work has been supported by competitive fellowships and grants from prestigious bodies such as the Royal Society, Wellcome Trust, and Generalitat Valenciana, with additional support from European collaborative research initiatives. In addition to her scientific endeavors, she is a dedicated mentor, guiding both undergraduate and postgraduate students at ULPGC. Her mentorship has been valuable in training the next generation of scientists, equipping them with the skills needed to thrive in academic and applied scientific research. Beyond her laboratory contributions, Dr. Muñoz Descalzo is deeply committed to science outreach. She is the director of DrosAfrica, a registered charity that cultivates a connected community of Drosophila researchers in Africa. Her dedication to public science communication is further exemplified in her accessible publications for the conversation and educational animations on developmental biology concepts, available on YouTube, which have engaged thousands of viewers. These initiatives underscore her belief in making science accessible and fostering a passion for research among diverse audiences. With her recent appointment as Sub-Director of the Advanced Confocal and Electronic Microscopy Service at ULPGC, Dr. Muñoz Descalzo continues to lead and innovate, driving advancements in biomedical research while nurturing a collaborative, inclusive environment for scientific inquiry. Her career embodies a unique blend of scientific excellence, educational dedication, and societal impact, establishing her as a transformative figure in the field of quantitative developmental biology. As we gather to learn from her insights and experiences, we are privileged to hear from someone whose work is shaping the future of our field. The floor is yours.
Okay. Thank you for the very kind introduction. It's really amazing to see all this in in place. Let me share. Can you hear me?
Yes, we can.
Okay, perfect. So again, thank you so much for the very kind introduction. And first, I want to thank obviously all the organizers and everyone that is there interested in my talk. Today, I thought I would be presenting the work that we've been doing in my lab recently, which is about how flies and mice can help us to better understand diabetes and Parkinson's disease. So, as I said, I want to give my talk in two parts. In the first one, I'll present you the latest results that we generated in the lab regarding how diabetes and our lifestyle can impact on fertility. This is work done in mouse, which is my favorite model organism. But I do have strong links with Drosophila labs because of my academic education, and I explain about it a bit later. And I will present you the work that we did in collaboration with a fly lab in Valencia, in Spain, showing how there is a link between Parkinson's disease and diabetes. And as I said, using both fly and mouse work. The diabetes work was done in the lab by a very talented PhD student. Now he moved on to be a postdoc in Janilia Farm in the US. He is Joaquin Lilagazon, and thanks to him, we published this paper last year on how maternal age, obesity, and hyperglycemia are associated with a delay in implantation development in mouse. So as a brief introduction, we all know that there is an issue, there's a big issue in the world regarding obesity. These are data from the whole world to show how obesity, and dark blue shows where we have a higher issue with obesity in the world. And we can see the most, so in Europe, in States, in Canada, in South Africa. In Africa, it's not really an issue in sub-Saharan Africa, but it is in Northern Africa. And how is it diabetes in the world? We have many people suffering from diabetes in the world. Again, mainly in Europe and in North Africa, but it is expected that it will increase 134% in the coming years, also in Africa. So it's really an issue, and it's really a problem, because there are so many diseases associated with diabetes and obesity that will impact in our future health. In Rwanda specifically, how is it with diabetes? It's true that it was not much of an issue in the early 2000s, but as you can see, there is an unexpected progression of the incidence of diabetes in Rwanda. And regarding fertility, how is fertility in our world? I'm showing here data from Europe, because it's where we have one of the main issues, especially around the Mediterranean area. And we can see that the age by which we have our first child has increased from the late 70s. It was around 26 years old. And now we have our kids almost in our 30s. In Spain, actually, we have a higher issue, as we tend to have our kids when we are in our 30s. And we know that maternal age has consequences on reproductive capacity, embryo quality, and even offspring overall health. And if we have our kids later, we tend to have fewer kids, which obviously results in an aging world population. You guys in Rwanda, there is not much of an issue. You have your kids at a very healthy 23 years old, and yeah, it's not much of an issue. But if the projections and are evolving as the demographic scientists think, it will probably increase in the coming years.
Dr. Silvia,
Yes.
the screen is not in presentation mode.
It's not?
Yes, ma'am.
Okay, you should have. How come?
You can try it again.
Is it now? Can you see it?
Yes, but it's not yet still in presentation mode. I can see the slides, the PowerPoint.
Well, maybe I can just do it this way.
Okay.
Yep. It's not
Yes.
what I wanted to do, but I mean, I think it's too late. Okay, so how is fertility and diabetes connected? We know that women with obesity and with diabetes are more likely to suffer from infertility due to menstrual irregularities, decreased quality, and there is a failure in uterine implantation of those embryos. Actually, most of the women with obesity and with diabetes, they have to go through assisted reproductive technologies in order to become pregnant. Here in this bit here, I'm showing the effect of how the pregnancy rate decreases as BMI, body mass index, increases in the people around in their 20s. It's this curve here. We can see that the higher their BMI, the lower the pregnancy rate. And this is so for all ages of the women. I'll come back to this line later. If we look at the 25 BMI index, which is the one that is considered normal and healthy, is this line here. We can see that, as I mentioned earlier, that as we age, our pregnancy rate, even if we are healthy or we have a healthy BMI, the pregnancy rate is decreasing. So how is our lifestyle impacting on our health? So we know that our lifestyle is not healthy. We have an increase in overweight and obesity. This leads to an increase in metabolic syndrome, in diabetes, in the incidence. We have a decreased fertility, not only because of obesity and diabetes, but also due to our advanced maternal age when we start having our kids. And finally, and this will be important for the second part of my talk, we know we have an aging world population, which will result in a massive increase in neurodegenerative diseases. So as you very well presented when you were introducing me, my lab is interested in developmental quantitative biology. What does this mean? What are we interested in? So we are interested in how cells make decisions, how from a fertilized oocyte we can get all the different shapes and cell types that we get in a baby. Not only within the baby, but also all those tissues that support their growth, like the yolk sac and the placenta. And the way we want to address these questions is quantifying. And so one of the other questions that we have in the lab is how can we quantify these decisions that cells make during embryonic development? How can we get quantitative data so that we can have or generate models that can help us to understand these decisions? The model system that we use in the lab is early mammalian pre-implantation development, which happens obviously before the embryo implants in the mother uterus. This is a cartoon showing development, mouse development. So in the pre-implantation development finishes around 4.5 days after fertilization, and then post-implantation continues. And specifically, the way that it happens is during the first day, we generate several cells, which are all equivalent, and each of them would be capable of developing a new mouse if it were transplanted into another female. The first decisions that cells make, let me remove this, is the cells in the periphery of these zygotes. They become trophoectoderm cells. These cells will form later the embryonic portion of the placenta, and they express transcription factors like CDX2, or another one is GATA3, for example. Half a day later, the ICM, the cells that are in the middle, they are the ICM cells. They do not contribute to the placenta, and these inner cell mass cells, they co-express markers like NANOC and GATA6. As I was saying, half a day later, these ICM cells, they are pushed towards one side because the trophoectodermal cells pump fluid so that they form this cavity. This structure is called blastocyst. And then the next decision that these ICM cells have to make is to become embryo proper, so to form all the cells, so to become those cells that will form the embryo, or become primitive endoderm cells, which will form later the yolk sac. Those cells that form the embryo proper keep the expression of NANOG and downregulate the expression of GATA6, and those cells that will form the primitive endoderm downregulate NANOG and keep the expression of GATA6. Then these cells sort, and at 4.5 days after fertilization, the mouse embryo implants. In humans, the process is pretty much similar. The difference is the timing, obviously. We have a more extended pregnancy, and implantation happens around between days 7 and 10, roughly. So in my lab, the way we want to address how cells make decisions is to focusing on these decisions, how cells from the nasal mask that co-express NANOG and GATA6 differentiate into epiblast cells or primitive endoderm cells. The way we do it from the quantitative point of view is we stain the mouse embryos using specific antibodies against NANOG and GATA6. We take confocal images like the ones I'm showing here, and then we use a software, specifically designed software called MINS, which allows us to understand each of the, to identify each of these cells, each of these nuclei, and then from these identified cells, it gives us information about the expression levels of each cell in each of the channels that we are staining. Okay, let me. So, what was the hypothesis we wanted to test in the lab together with Joaquin, with this PhD candidate? So the hypothesis was that maternal age and or metabolic syndrome, diabetes, would affect preimplantation embryo development, and this would have an effect in the fertility of these females. The way he addressed this question was to have several groups of mice to model age and how age affects fertility. He had different he had mice females of different ages, young ones, mature ones, which are equivalent to the perimenopausic age in women, and all females that were 12 months old that are menopausic, I mean, equivalent to menopausic women. This group was fed normal diet, so we were modeling only age, and then to model obesity and diabetes, he was feeding females of the same ages with a high-fat diet. And then on all these groups, on the 6 groups, he assessed obesity, diabetes, and then fertility, using both the traditional methods that are used in fertility clinics, together with the quantitative biology methods that we developed in the lab. Regarding obesity assessment, what he did was to weight the females over the period of 8 weeks, which was the period by which he was feeding the mice. And if we focus, so we have blue for the young, pinky for the mature, and black for the old ones. As you can see, the mature and old ones with normal diet, they didn't increase in weight. However, if he was feeding them with a high-fat diet, obviously they were gaining weight, and their final weight was really increased when compared to their equivalent age controls. Something very interesting happens with the young group. As you can see, there is no difference in the weight between the normal diet and the high-fat diet. And we associate this to the fact that these females of this age, they were undergoing their physiological growth. So regardless of feeding them with high-fat diet, they were not gaining more weight than their counterparts. Another way of monitoring weight, or another measurement that clinical doctors do, is to assess the body mass index. And as you can see, obviously, that we can already expect from the weight monitorization, there is not a change in the body mass index in the young females, but it is there in the mature and old female groups when fed with a high-fat diet. The other thing that he did was to assess the whole body composition. For that, he used a mini-spec lean fat analyzer, which is similar to an MRI, more or less. So this machine, what it allows us is to evaluate the body composition regarding fat, lean, water, and other tissues. So if we check fat and lean, what we can see is that the high-fat diet increases the fat content in the high-fat diet fed females when compared to their same age controls. Also, in the young group, in which we didn't observe any change in the weight or in the body mass index, and this was reflected as well by a decrease in the lean content. We can see that the lean content under high-fat diet in young, mature, and old females, there was a decrease in their lean content. So even though we didn't see any differences in the young group here, when we assess the body composition, we can obviously see it. This was telling us or suggesting that really the BMI that is usually monitored in the doctors, it shouldn't be really used, because we can have an increase in fat and it's not being observed in the weight. So how did he assess whether these females had diabetes? What he did was an oral glucose tolerance test. It's pretty much the same as we get as patients when the doctors want to assess whether we are diabetic or not. We feed glucose to the females, and then we measure the glucose content in blood over a period of time. We focus on the blue line here, which is the normal controlled young female ones. What happens is when you feed, the glucose, we have a peak in the glucose levels, and then it recovers due to the action of the insulin. If we look at the high-fat diet fed females in the young, in the mature, and in the old, we can see that it doesn't recover as quickly and as well as as it happens with the young females or the control females. Another way of showing this is to measure the area under the curve. And as we can see here, it's obvious that the area under the curve is higher for the high-fat diet fed females, indicating that glucose metabolism is altered under high-fat diet. For the fertility assessment, we used, as I said, traditional methods. These traditional methods are based on monitoring pre-implantation embryo quality by observational means. So basically what we do is we flash the pre-implantation embryos from young normal diet, high-fat diet, and mature normal diet, and mature high-fat diet. From the old group, we didn't get any embryos, which is kind of expectable, as they were already too old to mate, and it was different. So for the next experiments, I'll show only the young and mature groups. So if we look at the quality of the embryos in fertility clinics, they are classified as A, B, C, and D categories. A group are those embryos that look really, really nice. They are usually used, A and B are usually used for transferring into the women, because they are so good that they have higher chances to implant and to become a baby. We can see them here in the young normal diet embryos. These ones here, they look really nice. They are blastocysts. They have expanded. And then the quality C and D, they are really poor quality embryos. They are not usually used for transferring into females, into women. And we can see them very clearly here that even though there are some that look nice, there are many that are looking dodgy and don't really have a healthy morphology. When he measured, or when he quantified these embryos and put them into categories, we can see that the embryo quality is affected by high-fat diet, but only in the young ones. And we had fewer good quality embryos and more bad quality embryos. but we didn't observe any difference in the mature groups. And we think this is due to the fact that once you are old, your embryos are so poor in quality that even the high fat diet wouldn't affect their quality towards being worse. Then we went ahead and did the fertility assessment using our quantitative methods that we developed in the lab. So for that, what we did was to get embryos from the young normal diet, young high-fat diet, mature normal diet, and mature high-fat diet, and stain them for markers for epiblast, for primitive endoderm, or placenta, and then measure the expression levels and see how they were performing and how they were developing. This is the analysis. Sorry that it's a bit crowded, the slide. I was expecting it to show it in bits. So what we are showing here, and I want you to focus on this side of the slide here, is the number of cells that we have in each category co-expressing both markers, NANOC and GATA6. These would be cells that are still undecided. They are still in an ICM cell state. Then in reddish here, we have those cells that are committed to be epiblast and cells that are committed to be primitive endoderm, so the yolk sac and the embryo proper. And if we see how these cell fate decisions are being affected in the different groups, we can see that there are more cells in an uncommitted state, especially in the mature high-fat diet embryos, and fewer cells committed to the respective fates. And this is telling us that the embryos developed under high-fat diet or for mature females, they are delayed in development. I want to remind you that by morphological features, we didn't see any, by traditional methods, we didn't see any difference between mature normal diet and mature high-fat diet fed female embryos. But here we do see this difference, massive difference between those two categories, with more embryos being delayed in their development. Now I want to move on to the second part of my talk, which is the link between Parkinson's disease and diabetes. And this is work done in collaboration with Nuria Baricia, here in the picture. She was my PhD supervisor almost many years ago. She is based in the University of Valencia, and she is still working on Drosophila, or using Drosophila as a model system. And we did study the link between Parkinson and diabetes using her fly models and our mouse model with a high-fat diet fed females. So how is PD, Parkinson's disease, in the world? Again, we have an issue in Europe, in the US, and in Canada. Not so much in Africa or in Rwanda. Actually, in Rwanda is one of the countries where we have very few deaths due to Parkinson's disease. But it is known that diabetes is a risk factor to suffer from PD. In this systematic review and meta-analysis using 32.5 million participants with over 50,000 people suffering both from PD and diabetes, it was shown that suffering from diabetes was a risk factor to develop Parkinson's disease. And do remember that in Rwanda, it is expected that diabetes is going to increase. So the incidence of Parkinson's disease will likely increase as well. So what is PD? Everyone has heard about it. It's a debilitating disease, usually suffered by older people, and it's these people present motor symptoms like resting tremor, postural instability, bradykinesia, muscular rigidity, and also non-motor symptoms like mood alterations and sleep disturbances. it results in a progressive reduction in the quality of life, and patients require increasing care, being a burden for the society and for the family, obviously. It is caused by the loss of dopaminergic neurons in the substantia nigra pars compacta, and in this cell loss is due to cell death, because in these cells they accumulate protein aggregates called Lewy bodies. Nuria has been studying Parkinson's disease using Drosophila for over 20 years now, and she has been performing high-fast throughput chemical screens to screen for therapeutic compounds, because nowadays there is not really a cure for Parkinson. It's only there are only treatments to alleviate the symptoms, but not to cure it. And she has also been doing metabolomic analysis, large-scale metabolomic analysis, so that she could identify therapeutic targets for pharmacological intervention. For that, she has been using Drosophila models, Drosophila models of Parkinson's disease, and the one that she mainly uses is one fly model that she generated many years ago, based on the DJ1 beta inactivation, which is an ortholog of a gene which causes familial Parkinson's disease. These flies they present phenotypes similar to Parkinson patients. So how can we model in flies and in mice diabetes and PD? So to model diabetes in mice, I've just showed you, we can use the high-fat diet, and we will use diabetes in the flies. And in wild-type flies, to induce diabetes, we can feed them with sucrose. And to model PD, what we can do is to feed the fly, rotenone, so that this rotenone results in cell death of the dopaminergic neurons. And we can also use the DJ1 mutant flies that Nuria developed in her lab many years ago. We use this model to assess the link between Parkinson's disease and diabetes. So what are the metabolic alterations in these flies? So if we feed sucrose to the flies, obviously you have an increase in the glucose metabolism. There's an increase in the glucose content in the flies. But also, if we have fly models of PD, there is an increase in the sugar content in these flies. So there is a link, or we can see that glucose metabolism is affected in PD flies. The other thing that we can assess in the flies is to check their survival. So what she did in the GDJ1 mutant flies was to assess how many flies were alive after a period of time when compared to the control. And we can see that PD flies die at an earlier age than the wild-type ones. Also, when we feed the flies rotenone, we also see a decrease in survival. This is work done in a Biologist lab. I think some of you may know him in Nigeria. You can see that feeding, this is the control line, and this is the rotenone-fed flies. They have a decreased survival, but also in the diabetic, so-called diabetic flies, we observe that there is a decrease in their survival when compared to the control ones. So how is the motor performance in the flies with diabetes? Let's focus first on the PD ones, just to show you the control situation. So the way to assess motor performance is to do this climbing assays. So basically, we put the flies in a vial. We tap the vial so that they all go to the bottom, and then we record how much time it takes them to climb back up. In the PD, in the DJ1 mutant flies, we can see that their climbing ability is decreased when compared to the control flies. With rotenone-fed flies, we can see the more flies are kept or stay in the bottom of the fly, and they cannot really climb up like the control ones. But also with diabetes, if we focus here on the old flies, we can see that many of them, they have an issue in their motor performance. As you can see here, in the younger ones, we don't see much that effect, and we think that this is due to the extra energy that the sucrose is giving them, to have more energy. And what about the specific dopaminergic neuron loss in these flies suffering from so-called diabetes? The way to measure the activity or the amount of dopaminergic neurons is to measure the TH expression, which is a marker for these neurons. We can see that those flies fed with sucrose, they do have a decreased TH expression, reduced dopaminergic neuron content. The same way we see the same effect in our mouse model for diabetic. We see a decreased expression of TH. Finally, the last experiment that we did was to feed the diabetic flies with metformin, which is a well-known anti-diabetic drug. And then what we did was obviously to check that metformin, even though it's a human drug, it was working in the flies. We can see that it decreases the glucose, the carbohydrate levels in the flies. But it also induces the motor performance and recovers the relative TH expression, indicating that metformin can improve PD-related phenotypes in the flies. So just to summarize my talk, I've told you today about how our lifestyle and diabetes impacts on fertility using the mouse model, and how there is a link between Parkinson's disease and diabetes using both the fly and mouse work. I want to acknowledge the people that participated in this work. Obviously, Joaquin, he was a PhD student, a PhD candidate in my lab for a number of years. Now he moved on to Janilia Farm, as I said. The diabetes was advised by Ana Wagner and Geray Brito in our institution. The Parkinson's disease and the fly model is from Nuria Aparicio lab, together with her collaborators Cristina Solana and Franz Sanz. And with Sabine Fischer in Germany, we do the theoretical model of our results. And I think that would be from my side, and I'm happy to take any questions that you might have. Let me see if I can.
Thank you very much, Professor Silva, for your wonderful presentation. Those of us that are joining very lately, let me just run through the summary of what she talked about. The title of our presentation is From Flies to Mice, a study on diabetes and Parkinson's disease. She did talk about how our lifestyle can influence fertility. So the research was on the use of mice to see the link between diabetes and obesity by feeding the mice with high fat diets to see the effect of fertility. As we know, or as she rightly pointed out in our presentation, diabetes and obesity can be linked with menstrual irregularities, decrease over quality and fertility, resultantly. This obviously would lead to decrease in fertility, and also due to increase in, that what you have mentioned leads to decrease in fertility, as well as the increase in neurodegenerative diseases recently. So she did talk about the laboratory of development in terms of qualitative biology. The research that was conducted together with our PhD students. They did look at the early mouse embryonic development using NANOG and GATA6 and GATA3 gene expression levels. So they inducted or induced obesity and diabetes. And also, in the second half of our presentation, she did talk about the link between Parkinson and type 2 diabetes as a predisposing factor for, sorry, she talked about diabetes as a predisposing factor for Parkinson disease. And this was done by inducing flies, Drosophila melanogaster, uh, uh, by it was done by feeding them or introducing rotinol and sucrose to induce Parkinson disease and diabetes respectively in flies. And I must say that this is very inspiring. I don't know if that is summary of what I was able to capture from our lecture. I don't know if we have questions so that we can present to our guest lecturer.
Very nice summary.
Thank you very much. Okay,
Um,
questioner is asking, oh,
yes.
do you want to?
Yeah.
Someone is asking here.
What are
Okay,
the
go
most
ahead, Prof.
promising areas of research where the findings from Drosophila and mice could potentially be applied to improve patient outcomes from individuals with Parkinson's or diabetes? This is a question from Kathy. Um, well, the next step that we want to take with the Drosophila and mouse work is precisely use the screens that Nuria Aparicio performed in her lab using the fly model, using different drugs, and she was seeing really nice and impact on improving Parkinson disease phenotypes in flies. And now we want to test them in the mice here in my lab. The trouble we are finding there is that we are not really getting the funding that we need to do so, but I think it's a very powerful method that we can do nice and quick experiments doing Drosophila work, and then we can transfer those results to mice. It's cheaper, as you know, it's quicker to work with Drosophila, and then only those ones that give promising results in Drosophila, we transfer them to mice. And this, we are focusing now on Parkinson rather than diabetes, but it's something that can be done likewise for diabetes. It's true that for diabetes there are more drugs that work, and it's not so problematic to treat diabetes as it is with Parkinson. And then the second question was, how can advancements in fly and mouse models help identify biomarkers for neurodegenerative disease in individuals with diabetes, potentially leading to earlier intervention and better patient outcomes? So one of the things that Nuria is doing with the flies is doing these metabolomic assays by which she analyzes all the proteins that are affected under the different models. In Parkinson, she has been doing already some work, and she has published some papers on that. We want to look into those markers in the mice as well, once we get the funding, of course. And obviously, if we are able to identify those proteins, those biomarkers, which appear associated with Parkinson or diabetes, obviously then we can transfer those biomarkers and assess in human patients whether it is the same or not, so that we could generate new markers that would lead to an earlier intervention, as you are suggesting. Can animal models of Parkinson's disease and diabetes help identify new drug treatments that could address both conditions simultaneously? Of course. I mean, the disease, they are different diseases, of course, but there is a link, and the link is mainly in both systems. The oxidative, there's an oxidative stress, and actually with metformin, the anti-diabetic drug, it was shown some time ago that another neurodegenerative disease like Huntington disease was actually improved in those Parkinson's disease patients that were taking metformin because they were diabetic as well. So there are drugs that can help both diseases at the same time. The other question, in what ways do Drosophila and mouse models of Parkinson's disease help us understand the impact of insulin resistance on neurodegeneration? Very good question. The thing is, the link between Parkinson's disease and diabetes, or this insulin resistance issue that some diabetic patients have, it's very early days. I mean, people have noticed this link, and there are some works trying to assess the molecular link and see how they are both related, but it's not yet, as far as I know, follow the issue of insulin resistance on neurodegeneration. There are not many works that I know of studying that specifically. But obviously, it's a way of addressing the question using mouse or Drosophila models, so that we can understand better the system. How do oxidative stress and inflammation, common in both diabetes and neurodegenerative diseases, manifest in Drosophila and mouse models, and what implication does this have for potential? Adedeji is asking about potential therapeutic target through the link between Parkinson's disease and diabetes. Not yet. These are very early days. So for the treatment of Parkinson's disease, as I showed you, and we published it, we can see the metformin, a typical anti-diabetic drug. It does work to improve Parkinson's phenotypes in flies. Of course, one of the things that can be done as well is, once we assess the new Parkinson treatments that we want to assess in both in flies and in mice, then what we can do as well is obviously try to see whether they improve the diabetic phenotype. But yeah, I'm afraid the answer is not yet. Hello?
Yes, we can hear you, ma.
Hello?
Hello.
I'm back. I don't know what's going on, but I keep on being disconnected.
I think that's all the questions.
Okay.
Okay. Thank you very much, Prof, for your wonderful presentation. Um, I must express our heartfelt gratitude to you for your thought-provoking presentation, and I must say that your insightful scientific talk has actually opened a new horizon for all of us. And also, I believe that when next we call upon you to give us something like this again, you wouldn't hesitate to come to give us or share with us from your wealth of knowledge. Thank you very much for
Thank you.
giving us, sharing your time with us, and also to the organizing committee, the backroom staff, Mr. Aziz Adedeji, the MD herself, Professor Adedeji. I must say a very big thanks to you for your dedication and your meticulous planning, which is actually leading to this resounding success, as we can see in this seminar. And to all participants and attendees, thank you very, very much for your active engagement. For those of you that have asked questions, thank you very much. Let us ensure that whatever we have learned from what you have said today, we take it to our respective scientific communities so that to benefit our community at large. And I hope that when next, I hope to see you in our future programs as well. Thank you very much. I don't know if MD wants to add anything before we close the seminar for today.
Thank you very much. I just want to pass an information that our next webinar will take place on the 9th of December, and we will be calling our guest speaker is going to be one of the leading scientists in Rwanda, that is in person of Professor Leo Mutesa. So let us stay tuned and attend in mass for this coming program. Thank you all for your support and for trying to grace this webinar. Thank you.
Thank you.
Thank you very much. Bye-bye.
Bye.
Bye. Thank you. Bye-bye to you all. Thank you.